Good morning, everyone. Next sense that we are going to start is the olfaction. So, olfaction is sense of smell. Okay. So, the presentation has the contents. That is, first one is definition of olfaction. Second is anatomy of olfactory receptor. Third is physiology of olfaction, and lastly the olfactory pathway. So if you consider the contents of it, all the four special senses, they will have similar type of contents in the presentation. So olfaction definition, it is a sense of smell, and humans, we can recognize about 10,000 different odors, and this uh, to make this possible, our nose contains 10 million to 100 million receptors for the sense of smell or olfaction. See the number of receptors which are present uh, for the olfactory system or olfactory sense. It is much more greater than that of the gustation. In gustation there were only 10,000 taste buds, but in case of olfaction there are 100 million to 10 million to 100 million receptors are present. Okay, so these receptors are contained within a region called as olfactory epithelium, and the olfactory epithelium, it consists of three kinds of cell. That is, first one is olfactory receptor cell, second one is supporting cell, and the third one is basal cell. If you see the type of cells which the olfactory epithelium consists of, it is similar to that of the cells that form the taste bud. Taste bud also have the statutory receptor cell, supporting cell, and basal cell. Only thing that differs over here, it is olfactory receptor cell. Otherwise it is all the same, and function wise also it is almost same. I won't so it is exactly same, but it is almost same. So let's see anatomy of olfactory receptor. The olfactory receptor cells are the first order neurons of the olfactory pathway. So each olfactory receptor cell is a bipolar neuron. So what is a bipolar neuron? uh, it has two poles. Uh, like if the cell body, it is present at the midline, and on the both side it shows presence of dendrites. Okay, so it is like this. I will show you the structure, something like this. Pardon my drawing again. So at the center we will have cell body, and at the ends we have the dendrites. Okay, so this is bipolar neuron. Two poles are there. So in here we have the nucleus in here, and this is the cell body. Okay, so this is a bipolar neuron with exposed knob shaped dendrite. A knob means like the structure of dendrite is something like this. Okay, so the ends of it, they will have shape something like this, a knob shaped dendrite, and an axon projecting through the cribriform plate that ends in the olfactory bulb. So what is this cribriform plate and what is this olfactory bulb, that we will see next, or we will see in the actual diagram. So extending from the dendrite of the olfactory receptor cell are several non-motile olfactory cilia. In case of the taste bud, there were microvilli. In here we have the cilia. So microvilli, they are like small hair-like projection or finger-like projections, and these cilia, they are like more longer versions of the hair. So they are hair like projections only, but they are somewhat longer in length as compared to the microvilli. Okay, so these are the sites where the olfactory transduction means what? It is conversion of chemical signal to the electrical signal. Okay, so what is chemical signal over here? That in case of the olfaction, it will be the odorant that is responsible for the activation of receptor. So odorant as such is a chemical dissolved in a fluid which makes contact with the receptor present on the dendrites of the bipolar neuron, where the activation of the receptor leads to the generation of action potential in the first order olfactory neuron. Okay, so within the plasma membranes of the olfactory cilia, we can find the olfactory receptors, and which are responsible for the detection of inhaled chemicals. Okay, so the chemicals that have an odor that bind to and stimulate olfactory receptor in olfactory cilia are called as odorants. Okay, so olfactory receptor cells respond to chemical stimulation of odorant molecule by producing a generator potential, thus initiating olfactory response. So when odorant binds to the olfactory receptor, it causes generation of generator potential, which in turn leads to generation of action potential. Okay, and this generation of action potential in turn is responsible for the initiation of the olfactory response. So next structure is supporting cells. So supporting cells are the columnar epithelial cells of the mucous membrane lining the nose. Okay, so they provide physical support, nourishment, and electrical insulation for the olfactory receptor cells, and also help detoxify chemicals that come in contact with the olfactory epithelium. So what are the different functions of the supporting cell? Is that to provide physical support, then second is to provide nutrition or nourishment, then third is to provide electrical insulation to the olfactory receptor cell, and the fourth is to help detoxify chemicals. Okay, then next structural feature of the olfactory epithelium is the basal cell. So basal cells, as in case of the gustation, they are the stem cells which are located between the bases of the supporting cells. So they continually undergo cell division to produce new olfactory receptor cells, which live for only a month or so before being replaced. So here it differs from the sense of gustation. In gustation, the basal cells, they convert in, first of all they convert into supporting cells and then to the gustatory receptor cell. But in case of olfaction, these basal cells, they continuously divide to form the olfactory receptor cells. They don't form in between the supporting cells. Okay, so next structural feature of olfactory epithelium is olfactory glands, or also called as Bowman's gland. So what they do, they produce mucus that is carried to the surface of epithelium by means of ducts. So when duct is present, we the type of gland which we are talking over here, it is exocrine gland. Okay, so what is function of mucus then? It moistens the surface of olfactory epithelium, and it dissolves odorant so that transduction can occur. Okay, in case of gustation, it was saliva that was responsible for the dissolving the testin. Here, the mucus produced by the olfactory gland plays the role of saliva in case of the olfactory system. Okay, so both supporting cells of the nasal epithelium and olfactory glands are innervated by parasympathetic neurons within branches of facial nerve, which can be stimulated by certain chemicals. Okay, so impulses in these nerves in turn stimulate the lacrimal glands in the eyes and nasal mucous glands. Okay, so the result is tears and runny nose after inhaling substances such as paper or the vapors of household ammonia. Okay, so एकदम तिखट फोडणी वगैरे टाकली की आपण if we smell that thing, uh, we have the tears in our eyes. That is because the nerves that connect, or nerves that arise from the olfactory epithelium, they also connect to the lacrimal apparatus of the eye. So lacrimal apparatus of the eye is responsible for lacrimation. Okay, lacrimation is nothing but tears, formation of tears. Okay, and also these nerves, they supply to the nasal mucous glands. Okay, so when we smell something of strong odor, suddenly we will have tears in our eyes, as well as we have runny nose. Okay, so this is the structure of the olfactory epithelium. So you can see the cilia over here. This is the olfactory olfactory receptor cell. Then we have the basal cell over here. Then we have the Bowman's gland or olfactory gland, which is responsible for production of mucus. And this olfactory receptor cell then forms the olfactory nerve fibers. Okay, so these are all odor molecules which make contact with receptors which are present in the plasma membrane of cilia. Okay, and in between these cells, that is olfactory receptor cells, we can find the supporting cells. Okay, so physiology of olfaction. So let's see, binding of odorant to olfactory receptor protein in olfactory cilium, it stimulates membrane protein called as G protein. So understand this very clearly. The receptor which is present in the plasma membrane of cilia of olfactory receptor cell, it is of type G protein coupled receptor. Okay, so the odorant makes contact with the receptor which is associated with the G protein. So the G protein in turn activates enzyme adenylate cyclase to produce a substance called as cyclic adenosine monophosphate, which is termed as in short CAMP or CAMP. Okay, so this CAMP, cyclic adenosine monophosphate, in turn opens up sodium ion channel that allows sodium to enter the cytosol. So when we say sodium to enter the cytosol, it essentially means that the sodium, it moves from ECF, that is extracellular fluid, to ICF. So cytosol is nothing but ICF, that is intercellular fluid. So it causes depolarizing generator potential to form in the membrane of the olfactory receptor cell. So in last lecture only I have told you, when the sodium moves from exterior to interior of the cell, it leads to the depolarization. So same thing applies over here, but it is the secondary messenger like CAMP, that is cyclic adenosine monophosphate. It opens up the sodium channels which are present in the plasma membrane of cilia of the olfactory receptor cell. Okay, so if sufficient number of sodium ions the they move inside the cytosol of the olfactory receptor cell, then this depolarizing generator potential, it reaches the threshold. Okay, so and when it reaches the threshold, action potential is generated. So in order to action potential to generate, what is essential is that sufficient number of sodiums, they must move inside the olfactory receptor cell. If sufficient number of sodiums, they don't move inside the olfactory receptor cell, then there will be no generation of action potential. Okay, so this action potential which is generated, it is along the axon of olfactory receptor cell. That means it will flow through the axons of the olfactory receptor cell. So it is shown in this diagram itself that this is the cilia of the olfactory receptor cell, and in the cilia we have the receptor, odorant receptor, to which odorant molecule attaches. This odorant, when occupies the odorant receptor, it causes activation of G protein. Please don't look at this what is this gamma and beta, and what is this alpha. We are not concerned with that right now, but remember this odorant molecule causes activation of odorant receptor over here, and which in turn activates this G protein. Okay, and this G protein in turn activates adenylate cyclase. Okay, so once this adenylate cyclase gets activated, it converts or it causes formation of CAM, that is cyclic adenosine monophosphate, which is formed from the ATP. So adenylate cyclase, it is a type of enzyme that converts ATP, that is adenosine triphosphate, into cyclic adenosine monophosphate. Okay, so when this cyclic adenosine monophosphate which is formed, which is a secondary messenger messenger, this in turn causes sodium channel and other cation channel to open up, and when these channels open up, the cations, they will move from outside of the cell to inside of the cell, and when they move in sufficient numbers, it leads to action potential. Okay, so till the threshold is reached, this electrical impulse which gets generated into the cell, it is called as generator potential. But when threshold is reached, the generator potential is called as action potential, which then flows through, or which then travels along the axon of the olfactory receptor cell, and then it projects to the olfactory bulb. Okay, so olfactory pathway, let's see what is the olfactory pathway. So you can see over here, there is it is a cribriform plate. Over here you can see this bony structure which is present at the back of the nose, or at the superior portion of the nose, and at the ceiling of the nose we have the cribriform plate, and in this cribriform plate there are foramens present, that is the holes are present through which the these olfactory neurons, their nerves project to the olfactory bulb. So this olfactory bulb, it is actually present at the base of the brain. So this olfactory bulb which you can see over here, it is nothing but the this part of the brain, that is the base of the brain. You will find the olfactory bulb. Okay, so let's see what is the olfactory pathway. On each side of the nose, we have about 40 bundles of the slender unmyelinated axons of olfactory receptor cells which extend to about 20 olfactory foramina in the cribriform plate of the ethmoid bone. Okay, so these 40 or so bundles of axons collectively form right and left olfactory nerves. Okay, so the olfactory nerves terminate in the brain in paired masses of gray matter called as olfactory bulb. So what is olfactory bulb? It is the uh, masses of gray matter which is because formed because of the olfactory nerves. Okay, so these olfactory bulbs, they are located below the frontal lobe of the cerebrum, and they are present lateral to the crista galli of the ethmoid bone. So in order to understand this, what is this crista galli of the ethmoid bone, my suggestion would be, please refer to the anatomy of the skull, okay, from the chapter skeleton. Okay, so within olfactory bulbs, the axon terminals of the olfactory receptor cells form synapse with the dendrites and cell bodies of the olfactory bulb neurons in the olfactory pathway. So till the olfactory bulb, we have the olfactory receptor cell which is continuing. So one part of the olfactory receptor cell is responsible for the making contact with the odorant molecule, and the other part, or other part of the other end of the olfactory receptor cell, makes contact with the olfactory bulb neurons. Okay, so axons of olfactory bulb neuron extend posteriorly, that is to the back, and form the olfactory tract. Okay, so olfactory tract, it is made up of axons of olfactory bulb. So some of the axons of the olfactory tract, it project to the primary olfactory area of the cerebral cortex, which is located at the inferior and medial surface of the temporal lobe. Okay, so where we can find the primary olfactory area? In the temporal lobe. Okay, so the primary olfactory area, it is the location where the conscious awareness of the smell begins. That means whenever the impulse reaches from the olfactory receptor cell to the primary olfactory area, we can actually start feeling the, or we will have the sense of smell. Okay, so olfactory sensations are the only sensation that reach the cerebral cortex without first synapsing in the thalamus. So out of the four senses that we are going to study, the other three, that is gustation, vision, and the hearing, they all, the neurons of which will all first of all end up in the thalamus and then will reach to the cortex. But olfaction is the only sense whose neurons directly reach to the cortex instead of synapsing, or instead of reaching to the thalamus first. Okay, so other axons of the olfactory tract, it project to the limbic system and the hypothalamus. So I have told you in last chapter only that whenever the axons reach to this system, that is limbic system and the hypothalamus, it is associated with the emotional and memory evoked responses to the odors. So in case of the test also, how you can remember certain test, it is because of the neurons reaching to the limbic system and the hypothalamus. So similarly, in case of olfaction, similar thing will happen. Some of the olfactory neurons, they will reach out to the limbic system and hypothalamus, so that we will have emotional and memory evoked responses to certain odors. That means if you have faced, or if you know a certain, if you were using a deodorant like 10 years back, and suddenly on one day you were presented or you received a gift from your very close friend, and he gifted you that deodorant, and when you spray it, that time only you will know that I have used this deodorant uh, like some time back. Okay, how you know that? It is because of limbic system and hypothalamus. It stores those memories, it stores those odors, information of those odors, and by means of these these memories you can actually get acquainted with the odors which you have felt in the past. Okay, so from primary olfactory area, pathways they also extend to the frontal lobe, and an important region for the odor identification and discrimination is the orbitofrontal area. So from primary olfactory area, which is a part of the temporal lobe, some axons reach to the orbitofrontal area of the frontal lobe, where we there is a site of odor identification and discrimination. Okay, so how you can identify 10,000 different odors? It is because of information which is stored in the frontal lobe, and which part of frontal lobe? That is orbitofrontal area. Okay, that's how you can discriminate or distinguish between two closely related odors. Okay, so that is how the olfactory pathway works. Okay, and with this we have finished with the chapter, that is olfaction. Okay, thank you.